Hello students, welcome to EPG Pathshala. I am Dr. Vikas Jain from BS Anangpuria Institute of Pharmacy, Faridabad. Today I am going to talk about module Monophasic Liquids Formulation Consideration. The content has been taken from paper Product Development 1. This lecture will make you learn about importance of viscosity, preservation, appearance and palatability, and the product stability, packaging, storage and labeling consideration in formulation development of monophasic liquids. You are expected to be familiar with physicochemical aspects of drugs and formulation, which you might have studied under physical pharmacy and pharmaceutical technology. The solubility and stability factors can be approached with the precision long associated with the exact sciences. On the other hand, flavoring and other organoleptic characteristics remain subjective factors for which the application of the scientific method still plays a distressingly minor role. Thus, the successful formulation of liquids as well as other dosage forms requires a blend of scientific equity and pharmaceutical art. Formulation consideration generally includes physicochemical aspects of drug and formulations. It also includes formulation parameters relevant to product development. It involves basic principle of formulation at any scale, let it be small scale, at large scale, at industrial level or at R&D level. It helps to enhance acceptability and to bring elegance in the product. It also helps in designing a stable formulation. So, by employing all those considerations, a tailor-made approach can be developed to meet the individual needs. Now, first of all, we'll discuss the important parameter, that is viscosity. It is sometimes desirable to increase the viscosity of a liquid, either to serve as an adjunct to palatability or to improve pourability. This can be achieved by either increasing the sugar concentration in the formulation or by incorporating viscosity controlling agents such as polyvinyl pyrrolidone or various cellulosic derivatives. The example includes methyl cellulose and sodium carboxymethyl cellulose that we generally use during our practicals. These compounds form solution in water that are stable over a wide pH range. Methyl cellulose and carboxymethyl cellulose are available in a number of different viscosity grades. Because of the significant opportunities available for interacting with salts and other formulation ingredients, the viscosity control should be studied in the formulation and over the shelf life of the product. Carboxymethyl cellulose may be used in solutions containing high concentrations of alcohol up to 50% without precipitating. It is precipitated, however, as an insoluble salt of a number of multivalent metal ions such as statine, iron, and calcium. Methyl cellulose polymers do not form insoluble salts with metal ions, but can be salted out of solution when the concentration of electrolytes or other dissolved materials exceeds certain limits. These limits may vary from 2 to 40% depending on the electrolyte and the type of methyl cellulose involved. Viscosity inducing polymers should be used with a degree of caution. They are known to form molecular complexes with a variety of inorganic and organic compounds, and in doing so, it influences the activity of these compounds. It is conceivable that highly viscid systems that resist dilution by gastrointestinal fluids might impede drug release and absorption. Let's discuss various polymorphic forms that do affect solubility characteristics and the final formulation characteristics of monophasic liquids. Variation in lattice energies between amorphous and crystalline forms can significantly influence a drug's aqueous solubility, and increases of several hundredfold were observed for morphine and benzimidazole derivatives. Furthermore, a substance may exist in more than one crystalline form, such as chloramphenicol, progesterone, cortisone, prednisolone, etc. Polymorphic transformations routinely observed for pharmaceuticals are structural differences resulting from different crystal arrangements of molecule in the solid state. Although thermodynamic differences between polymorphs disappear once dissolved, solid state disparities in internal crystal structure between polymorphs may yield vastly different physicochemical properties, and changes may include melting point, chemical stability, solubility, enthalpy, dissolution rate, and color, etc. Freeze thaw as well as more elevated temperature cycling tests can provide a useful technique for evaluating crystal growth and crystal growth inhibitors. After viscosity, another important consideration is preservation. Liquid preparations are mostly non-sterile pharmaceutical products to be contaminated by microorganisms, marketed in a multi-dose form, enhancing the risk of exposure to microbes upon repeated dosing. Inclusion of sugars generally provide growth supporting substrates. The manufacturing process may also contribute to possible microbiological contamination. Therefore, it is essential that liquid preparations be protected against microbiological deterioration by adequate preservation. Preservatives are an essential part of every liquid formulation unless there is sufficient preservative efficacy in the formulation itself. Many drugs in solution are subject to oxidative degradation. Oxidation reactions are mediated by free radicals or molecular oxygen, often catalyzed by presence of metal ions. Antioxidants generally work by consuming oxygen at a faster rate than the drug substance itself reacts with oxygen. It protects drug substance until antioxidants component are fully used up. The use of antioxidants imposes a lag time upon the decomposition profile of the drug, enhances the preservation or shelf-life stability of the finished drug product. Preservatives are an essential part of liquid formulations unless there is sufficient preservative efficacy in the formulation itself, such as due to high sugar content, presence of antimicrobial drugs, or solvents that inhibit growth such as alcohols. An ideal preservative should have a broad spectrum of activity, physical, chemical, and microbiological stability, and physiological compatibility. There is some psychologic basis to drug therapy, and the odor, taste, and color of a pharmaceutical preparation can play an important role. An appropriate drug has its most beneficial effect when it is accepted and taken properly by the patient. The proper combination of flavor, fragrance, and color in a pharmaceutical product contributes to its acceptance. Although most drug substances in use today are unpalatable and unattractive in their natural native state, their preparations are presented to the patient as colorful, flavorful formulations attractive to the sight, smell, and taste. These qualities, which are the rule rather than the exception, have virtually eliminated the natural reluctance of many patients to take medications exclusively because of disagreeable odor or taste. The appearance or color of liquid products is often synchronized with the flavors used. For example, green or blue for mint, red for berry, and so forth. Because the amount of dyestuffs allowed in pharmaceutical products is strongly regulated, this presents problems, especially where there is a need to mask features of a preparation. The dyes and the maximum amounts permitted for use in pharmaceuticals vary from country to country, and each regulatory agency also revises its approved list from time to time. In some instances, solutions are made to sparkle by passing them through a filtration process. Often, adsorbents are used in the liquid preparation to remove fine particles, hence imparting a greater clarity to the solutions. Filtration often presents problem, but with the help now available from major filter manufacturers, most problems can be readily solved. Particulate matter may be introduced through lint and fibers from the solvent or trace quantities of insoluble contaminants in one or more of the formulation components. It is quite common, for example, for alcoholic solutions of natural flavors to precipitate pectins and resins on addition to the bulk aqueous solution. The removal of this and other particulate matter is referred to as polishing and technically may be accomplished in several ways. Those different ways are by settling and subsequent decantation, by centrifugation, and by filtration. Filtration is the only practical method when large volumes of liquids are involved. It is in order at this point, however, to mention some of the effects that this process can have on the product. The filter pads most frequently used for oral liquids were formerly composed either entirely of asbestos or of mixtures of asbestos and cellulose. With the finding that asbestos fibers can cause cancer, liquids are now filtered whenever possible through membrane filters. Studies should be carried out before and after filtration to determine the extent, if any, to which actives, preservatives, flavors, colorants, and other important product components are adsorbed. Production conditions should be simulated as closely as possible and particular attention given to filtration rate and liquid to filter surface area ratio. A much less common problem is that of extraction of materials from the filter pad. This is only of concern insofar as the extracted material may affect the physical and chemical stability of the product. Pharmaceutically acceptable filter pads and aids contain no biologically active components. For this reason, stability studies must be carried out on product made by same process as the one to be used for ultimate production. Once we have prepared a good product, it is acceptable, it is looking elegant, smelling good, tasting good. One important consideration that is also taken into account is drug product stability. Drug substances are generally more susceptible to degradation, as we all know, because of moisture content, sugar content, as compared to solid dosage form. As a class of formulations, oral liquids are more complex in composition than parenterals, and more interactions may occur affecting the stability of the product. It is therefore necessary to not only consider the solution stability of the drug, but also the stability of excipients such as colorants, flavors, preservatives, solubilizers, thickening agents, and sweetening agents. The techniques for predicting chemical stability and physical stability of homogeneous drug systems are well defined nowadays. The techniques for predicting the chemical stability of homogeneous drug systems are well defined. The formulation chemist should consider both the pH solubility profile and pH stability profile while selecting the optimum pH for formulation of the liquid oral dosage form. Physical instability of liquid formulations involves the formation of precipitates, less soluble polymorphs, adsorption of the drug substances onto container surface, microbial growth, and changes in product appearance. Evaluation of product acceptability is subjective and includes properties such as color, odor, taste, and clarity. According to the ICH guidelines for the stability testing, the design of formal stability studies for the drug product should be based on knowledge and behavior and properties of the drug substance. Photostability testing should be conducted on at least one primary batch of the drug product, and data from stability studies should be provided on at least three primary batches of the drug product. The primary batches should be of the same formulation and be packaged in the same container closure system proposed for marketing. The manufacturing process used for primary batches should simulate the process that will be applied to production batches and should provide product that is of the same quality and that meets the same specification as that intended for marketing. Two of the three batches should be at least pilot scale batches. The third one can be smaller if justified. Where possible, batches of the drug product should be manufactured using different batches of the drug substances. Stability studies should be performed on each individual strength and container size of the drug product unless bracketing or matrixing are applied. Other supporting data along with this can also be provided. Stability testing should be conducted on the dosage form packaged in the container closure system proposed for marketing, including as appropriate any secondary packaging and container label, etc. The testing should cover as appropriate the physical, chemical, biological, and microbiological attributes, preservative content, for example, antioxidant, antimicrobial preservatives, and functionality test, for example, for a dose delivery systems. Analytical procedures should be fully validated and indicating stability. Shelf life acceptance criteria should be derived from consideration of all available stability information. It may be appropriate to have justifiable difference between the shelf life and release acceptance criteria based on the stability evaluation and the changes observed on storage. Stability studies are necessary to support the storage and shipment of the drug product. These studies are done on packaged as well as unpackaged drug products. Typically, two types of studies are conducted. First one is thermal studies and second one is freeze-thaw studies. Thermal studies are done for all products by exposing the drug products to a few temperature cycles. For example, conducting 3 cycles where each cycle includes drug product first stored at 40 degrees centigrade for 4 days and at 25 degrees centigrade for 3 days, while freeze-thaw studies are done specially for liquid products by exposing the drug products to a few temperature cycles, that is, conducting 3 cycles where each cycle includes drug products stored at -10 to -20 degrees centigrade for 4 days and at 25 degrees centigrade and ambient RH for 3 days. A single primary stability batch of the drug product should be tested for antimicrobial preservative effectiveness in addition to preservative content at the proposed shelf life for verification purposes, regardless of whether there is a difference between the release and shelf life acceptance criteria for preservative content. In general, a drug product should be evaluated under specified storage conditions with appropriate tolerances that can test its thermal stability and, if applicable, its sensitivity to moisture or potential for solvent loss. The storage conditions and the lengths of studies chosen should be sufficient to cover storage, shipment, and subsequent use. Stability testing of the drug product after constitution or dilution, if applicable, should be conducted to provide information for the labeling on the preparation, storage condition, and in-use period of the constituted or diluted product. This testing should be performed on the constituted or diluted product through the proposed in-use period on primary batches as part of the former stability studies at initial and final time points. And in case of full shelf life, long-term data will not be available before submission at 12 months or at least last time point for which data will be available. In general, this testing need not be repeated on commitment batches. New drug application for every product requires a package interaction description. Obviously, final stability data are to be developed in the final package form only. Although glass bottles are fairly resistant to many products, caps and liners are often not. Even the integrity of the caps needs to be evaluated, applying exact torque in closing the bottles intended for stability evaluation. This is important to prevent cap breakage that might adversely affect stability. In order to enhance the shelf life or stability of a liquid dosage form, packaging plays a major role, or it generally enhances the shelf life of that particular product if it has been packaged properly. So, liquids generally supplied in multiple dose or single dose containers, and it should be packed in bottles sufficiently wide mouthed to facilitate the flow of the content. They are administered either in volumes such as 5 ml or multiples of 5 ml, or in small volumes as drops. Each dose of multiple dose liquid is administered by means of suitable measuring device, usually a dropper, that is generally provided with the container itself. In most instances, either plastic or aluminum cap are applied to the bottles. The liners used in the caps should demonstrate full compatibility with the product, including any adhesive used. Proper torque should be applied to ensure a tight seal. Pilfer evident packaging wherever used must comply with the regulatory requirements. A microbial survey should be performed on all packaging materials that comes into the contact with the product to ensure that microbial contamination is not introduced at this time point. Attention must also be paid to details during packaging operations. For example, on small volume orders in which bottle closures or tips for plastic squeeze spray containers are often placed on dye product by hand, this procedure can be a source of microbial contamination from the hands of operators, unless gloves are used that are pre-sterilized and periodically disinfected during the use. According to Indian Pharmacopoeia, Liquids or powders and granules for the preparation of liquids should be stored in well-closed containers at temperatures not exceeding 30 degrees centigrade. This is applicable for storage of all categories of oral liquids. Aromatic waters are to be kept in airtight, light-resistant containers. However, colloidons, that is a topical preparation, should be stored removed from fire as it contains ethanol and ether. Liniments should not be stored in polystyrene containers as it may cause certain incompatibilities. Let us discuss the labeling requirements for monophasic liquids. For oral liquids specifically that are supplied as drops, the label states the number of drops per gram of preparation if the dose is stated in drops, or the number of drops per milliliter of preparation if dose is stated in volume. For oral liquids supplied as granules or powders to be constituted before use, the label states that the contents are meant for preparation of an oral liquid. The directions for preparing the oral liquid, including the nature and quantity of the liquids to be used, the conditions under which the constituted solution should be stored, the period during which the constituted oral liquid may be expected to remain satisfactory for use when prepared and stored in accordance with the manufacturer's recommendation. The strength in terms of the active ingredients in a suitable dose volume of the constituted preparation. Labeling requirements for cutaneous preparation should include all those things that has been mentioned. The first thing is the names and concentrations of the active ingredients. It is intended for external use only. It should be shaken before every use. The date after which it is not intended to be used, the storage conditions, the directions for using the preparation, and any special precaution that is to be observed during its storage and uses. Increasing knowledge into personalized medicine has demonstrated the need for individual dosing. Pediatric and geriatric drug delivery also need individualized dosing, patient-adapted drug formulation, and delivery devices. Further, some drugs with small therapeutic window needs precise dosage adjustment. It is obvious that suitable dosage forms are urgently needed, enabling the selection and application of individual doses to transfer fundamental knowledge on personalized medicine into daily medical practice. In the best case, these dosage forms should be suitable for complete patient population, starting from young children to the elderly patients. Liquid drug formulations for oral drug administrations are available as solutions, syrups, emulsions, or suspensions. Homogeneous liquids like solutions or syrups with completely dissolved APIs show advantages over emulsions and suspensions as they ensure uniform doses when withdrawing single doses out of a multi-dose container. Single-dose containers for oral liquid medications could be sachet or stick packs, but they are currently not available on the market. Therefore, for accumulating dosing approach, only dropping bottles and tubes can be considered useful for current need. Partition dosing approaches require measuring or counting tools. It can be distinguished between tools which are part of the primary packaging, such as dropper inlets or dropping tubes, and separate dosing devices like dosing spoons, cups, dropping pipettes, and oral syringes. Out of all available modern dosing options, oral syringes are considered as the best dosing devices. So, students, let us now summarize what we have learned from this module. The first thing is, for successful development of a product, parameters to be decided in advance to meet product specific requirement. It should always be decided in advance. It may involve use of some additives, excipients and specialized procedure too. It helps in development of suitable container closure system, and it also helps in generating labeling requirements. A single drug can be formulated in multiple dosage forms to meet various requirements and can be applied to various sites too. Currently, there is a definite need for development of novel dosage forms and delivery devices for oral individual drug therapy. Thank you.